Welcome to the Watershed Watch training presentation on using the R-Card method for E. coli analysis. This intro should be watched along with the YouTube training video. To begin with, it's important to know why we analyze stream samples for E. coli levels. E. coli bacteria in the water serve as an indicator of fecal waste contamination from warm-blooded animals, including humans, livestock, pets, and wildlife. When present, they may be accompanied by pathogens that represent a human health threat, such as a greater risk of contracting a disease or an infection. When E. coli concentrations exceed a certain level, the waterways are noted as being unsafe for activities such as swimming, wading, boating, or fishing. Watershed Watch has traditionally used laboratory analysis of water samples to measure E. coli concentrations. As of 2024, we are encouraging the use of the Roth Biosciences R-Card method. This will eliminate the need to transport samples to lab locations, which can be far away, and reduce analysis costs. The cards are coated with a media surface that allows the bacteria to reproduce and grow in an incubator. The layers of the cards seal after the water sample is taken, making transport easier. No ice is needed to preserve the samples, and the bacteria are easy to count as blue dots once the cards have been incubated. For each stream sample, you will take 3 samples using the R cards. For each site you sample, you will need 3 cards and 1 pipette. You will also need access to an incubator, which can be shared among samplers, checked out or used at a support hub, or purchased or constructed for individual use. The information and cost for purchasing these supplies is available on the Watershed Watch website. Also on the website is a one-page guidance printout for the steps to take at your sampling location. You may want to print and laminate this card for quick reference. These are the basic steps of the field sampling process. Carefully unwrap the pipette from the bulb end and avoid touching the tip where you will take your water sample. Withdraw the 1 milliliter of water into the pipette from the stream without touching the stream bottom. Carefully lift the top flap of the R card and then dispense the water in the center of the grid on the card and lower the flap. This will be done with 3 samples of water for each site. Some practice with this method has led to these tips for success. First, we suggest using a clipboard or other flat surface to ensure a level surface for the card so that the water disperses evenly and does not run off the card. You may want to use binder clips to hold the cards in place if it's a windy day. As mentioned, avoid touching the narrow end of the pipette to prevent contamination from your hands, clothing, or the bottom of the stream. We suggest sampling a few inches below the water surface. Also, before sampling, make sure that you have looked at the 1 milliliter mark on the pipette. It is the one nearest the top or the bulb end. Once you've collected the sample, slowly squeeze the pipette to release the water onto the center of the card. Lowering the top flap will distribute the sample around the card. You should not use your hand or a tool to press down on it. Getting the water to the 1 milliliter mark can be a little tricky, so you may want to practice your technique with a spare or used pipette. Here are some hints about the technique that may help you obtain the 1 milliliter sample. First, squeeze the bulb of the pipette and insert it in the stream. Release some pressure on the bulb to withdraw more than 1 milliliter of water. Continuing holding pressure on the pipette bulb as you withdraw it from the stream. Then squeeze a little bit slowly to release the water gradually until it reaches the 1 milliliter line. Once it reaches the line, release the pressure completely on the bulb. It's okay if you see a little air bubble in this pipette. After you have placed the water sample on the card and lowered the flap, you should wait a couple of minutes for it to fully seal. Then place the 3 cards in a sealable plastic bag for transport. The cards do not need to be placed on ice, but they should be kept out of extreme heat. Avoid having them in direct sunlight or in a hot vehicle between sampling and placement in the incubator. If necessary, you can place the plastic bags of cards in a small cooler with a cold pack to keep them cool. You should plan to put the cards in the preheated incubator within 3 hours of sample collection. This is the one-page reference for the incubation and counting phases of the R-card procedure. You may also want to print this from the website. Here are a couple of tips related to the incubation. If you need more space for cards in the incubator, it's okay to stack them up to 15 on top of each other. Also, make sure that the thermostat on the incubator or a thermometer reading stays between 35 and 38 degrees Celsius while the cards are inside it. You can check it periodically to be sure. Here are some tips related to counting the E. coli colonies. First, use the grid on the card to methodically count and record colonies by row or column. Don't count the tiny dots that are smaller than the tip of a pen or a period of regular type font. Record only the dark blue or purple colonies. The pink colonies represent other types of coliform bacteria. When colonies are too dense to count, you can either estimate the number and multiply by the number of squares with a similar density, or if colonies exceed 200, you can record the final result as greater than 20,000 since you will be multiplying the value by 100. The University of Kentucky is working on an app that will enable you to submit a photo of your card for automatic counting. We are planning for this to be available later in 2024 to assist you with this step. You may also want to take a photo of your card and compare counts with a sampling team member. If you continue to have very high E. coli counts, you may want to consider one of these two options. You can try to determine potential sources of the E. coli by sampling at locations upstream of the site or at the mouth of tributaries entering upstream. You can also request follow-up lab analysis of your site samples by contacting your Watershed Watch Basin representative or one of the program coordinators. They will try to help you arrange to take a sample and deliver it to a lab for analysis using their E. coli measurement method. This slide is a review of how to complete the section of the data form pertaining to R-card analysis. You will need to record each of the fields on the form as you incubate and count the colonies. Then you will enter this data along with the other observations that you've recorded during your site visit. Here are some safety and cleanliness recommendations for you to keep in mind as you complete the R-card procedure. Wear disposable gloves for the sampling and cleanup steps. Avoid touching the bacterial colonies on the card. Wash your hands or use hand sanitizer after handling the cards. Keep work surfaces clean, and after you are finished, you can place the cards in a sealable plastic bag and microwave it for 4 minutes or swirl with a bleach solution before placing them in the garbage. This short video shows the steps involved in the R-card process. After watching it, please answer the questions in the R-card quiz for completion of this module. For in-person training sessions, here are the steps that participants should follow to practice and become more comfortable with the R-card method. Thank you.